Hi, my name is Lisa Massaroni. For the past summer, I've been working over in 320 Charles in the genome sequencing platform, more specifically the 454 team. And a little bit of background on sequencing. It used to be done with Sanger sequencing, a technology that could produce about 384 reads per run. And recently, Sanger sequencing has been replaced by newer technologies, which 454 is one of those newer technologies. And 454 sequencing technology can produce reads, it can produce about 1 million reads per run, so it results in a drastic decrease in the cost of a sequencing run. But one thing that you sacrifice with newer technologies is read length. So Sanger sequencing could produce average read lengths between 750 and 1,000 base pairs, whereas 454 sequencing is capable of producing 500 to 800 base pair read lengths, but we tend to see about 300 to 500, so it's below that range. So the goal of my project was to explore size selection methods, which is one of the steps in library construction that would optimize the read lengths in 454 sequencing. And the justification for our project was that we explored historical data and found a positive correlation between library size and average read length, which is shown here. So this graph which I made from data that I compiled shows along the bottom the library sizes, which is the length of fragments that you send to sequencing, and along this axis is the average read lengths that are produced from those libraries, and as you can see, the positive correlation. So our approach was we tested different shearing times and size selection methods, and then analyzed using 2 different methods. So the shearing that is standard in 454 uses a Covaris E210, which uses sonification to shear DNA into fragments. And what we chose to look at with shearing was different times. So the longer you shear, the shorter fragments you'll have. So we looked at 80 seconds, 100 seconds, and 120 seconds. Then we looked at 4 different size selection methods. The first is a SPRI, or solid phase reversible immobilization, which basically takes the DNA that you have in your sample and you add polyethylene glycol and salt, which if concentrations of the PEG, or polyethylene glycol and salt, are high, all of the fragments will bind, but longer fragments bind preferentially. So first longer fragments will bind, and if you don't add enough PEG and salt concentrations, only the shorter fragments won't be able to bind. So you can remove the shorter fragments remove them with the supernatant, and then you're left with only longer fragments. The next method that we looked at was a gel cut, which we ran our sheared fragments alongside a 100 base pair ladder on a gel, and then cut the range from 500 to 800, and used a QIAquick gel extraction kit to extract the DNA. And then the final 2 methods of size selection that we explored were called a Pippin Prep and a Caliper Lab Chip XT, which are newer technologies that we have here at the Broad. And they both work on this basic principle, where you load your DNA into a loading well, and on the other end there are switchable anodes. So originally the DNA is flowing toward a positive charge in the waste chamber, and then a detector, which works with an internal ladder in the machine, tells when 500 to 800 base pairs are flowing through, and it will switch and pull the DNA into an elution chamber, and you can just pipette out the DNA and it'll be the size range that you want. So then finally we utilized 2 different methods of analysis. We would have liked to sequence all of our samples, but it would have been too expensive and also taken longer than my 6-week internship. So for some of the samples, we did do a 454 sequencing run, which was able to produce actual data on average read length. And then for other samples, we simply used the Agilent 2100 bioanalyzer, which gives us an electropharogram, which tells how many fragments you have and how many, or the sizes of fragments that you have and how many fragments are that size. So we did 4 conditions that we analyzed with sequencing, which the standard 454 protocol is 120-second shear and an 0.7x spry. So we chose to analyze a shorter shear time, 80 seconds, and then a normal spry. Then we did the normal shear time and a tighter spry, a gel cut, which is the size selection method that they would have used in the past, as well as a pip and prep, which is one of the newer technologies. Then there were 18 conditions that we analyzed with Agilent, and it was 3 different shear times, 80 seconds, 100 seconds, and 120 seconds, 4 different spry concentrations, a dual spry, which is first a concentration that will select against larger fragments, then a concentration that will select against smaller fragments, and finally the caliper, which again is one of the newer technologies. So the results that these different methods showed us, for the conditions we analyzed with 454 sequencing, the results of library size and average read length didn't show a statistically significant difference between conditions tested themselves or between historical data. As you can see here, the gray is historical data and the white is our data. And our data falls right in the normal distribution of past runs. So although our data was not significantly different from past runs or from the conditions that we tested, this still was beneficial in that it showed us a gel cut, which they would have used in the past, the spry that we use now, and the newer technologies are all capable of producing average read lengths that we like to see. And then secondly, the conditions we analyzed with Agilent, we looked at yield, which was the total DNA, measured by the area under the curve of the electropharogram, selectivity, which was the percent of total yield that was between the 500 and 800 base pair range, and also omission of fragments under 500 base pairs, which was the percent of the total yield under 500. So if you look at the darker bar here, the standard is outlined in the darker shade. And for yields, it is relatively good. It's up in the higher range of things. So the standard protocol is good for yields. For selectivity, the standard was sort of right in the middle, but there were some conditions that produced a much better selectivity percent. For example, the 120-second shear and caliper produced 70% of the sample was in the range that we wanted it to be, and then also the shorter shear times with a dual spry produced very good results as well. Finally, omission. On this chart, you want as low a bar as you can possibly get because it's the percent that is under 500. So if you look at the standard, it is actually on the worst end of things. It's one of the higher bars, but there are ways to produce libraries with a better omission percent. Again, the caliper is very good, and also shorter shear times with tighter spries are very good. So the conclusions that we drew were that the standard 454 protocol that we use right now isn't necessarily the best for producing optimal average read lengths, but in order to truly validate those results, we would need to take the conditions that we analyzed simply with an Agilent and actually sequence samples that underwent those conditions to test if they do have the impact on average read length that we expect them to. And another direction we would like to take in the future is to further analyze the historical data that I compiled and see if there are correlations between other areas or conditions and average read length. So thank you for listening to how I explored size selection methods to optimize average read length in 454 sequencing, and have a great night.